Hi, welcome to PhDizzle. I'm your host, Alice Cheng, and today I am interviewing Dr. Jiaxuan Chen, who got his bachelor's in biological sciences and biomedical sciences from Peking University and his PhD in bioengineering and biomedical engineering from a combined program between Georgia Tech and Emory. And he's currently an assistant professor at Harvard. You are very accomplished in academia, but did you always know that you wanted to study biology and to be in academia?
Yes, I'm interested in biology from elementary school. I don't even know what that class called in America, but I think it's horticulture, maybe.
Like plants?
where their teachers tell you how to grow a little plants. I remember I grew a little potato in a pot, and I planted one piece of a cut piece of a potato with a sprout, and I harvest like 5 little potatoes.
Wow!
And in the fall, and I sent it to the class, and I guess I was the star of the class, which triggered.
Overachiever.
Some interest.
Yeah, like after the year ends, you still have the potato.
Yeah, actually, it's put in the a formaldehyde, and it's in the library of that elementary school. I don't know if it's still there.
Oh, but, that's so cool. Wow.
That's too far away. But I guess everybody, to a point of their life, a lot of people that love plants, small animal, fluffy animal.
Yeah.
I always like that, so that's keep growing. So I guess I should do biology for college.
That makes sense. And you went to Peking University, which is like the Harvard of China. And so not only is it really a great school, but the competition is also really crazy in China because there's so many people to get into school like that. What was that testing process and admission process like for you?
Yeah, it was really a lot of pressure for the college entry exam. A lot of people sort of depend on that exam to change their career path. Obviously, if you got into Peking University or Tsinghua University, you are set to a good start for your life, right? And there are just so many people trying to get into the top school. It's a little different, China to US. In US, you have like the Ivy League, which is a bunch of school, and you have even Stanford and other really, really good university that is out of Ivy League, but give you also good opportunities. So I think in China, there are just relatively less amount those really top schools, so that's why the pressure is so high. I was lucky because I attended an Olympic biology test, and I was scored like I don't know, it's like first class in Beijing in the city.
Wow.
So I was actually I'm exempt from the entry exam. So
Well, but you did a lot of work to do the biology exam, right? So it's like you still spent a lot of time studying, but it was kind of like for a different thing.
Yeah, and actually the school asked me to take the exam anyway, not because I needed it, because they wanted another good score, so they can market themselves. So I took that exam. And I was my score was qualified as well. So
just for fun. Wow.
That year is also the year of the COVID-1.
Was it US? SARS? Because we
called SARS.
Okay, maybe that's what. Okay, because I was like, I remember something, and then I was like, I don't remember COVID, but I guess SARS-CoV is the official name.
Yeah.
Oh, okay. Yeah. So did you have to like stay inside? Was it like quarantine the same way?
Yeah, it was. So we are not allowed to go to school to study, which is crazy consider how important this exam is, right? So everybody start study in their home for half a year until they go to take the exam. So everybody is somewhat unprepared. So it's It's a
Wow.
little crazy, yeah.
Wow. Okay. Yeah, I guess I didn't think about that because I guess SARS wasn't as big in the US, at least. Like it was kind of scary, but it wasn't like a national global pandemic.
No, it's not. It's not qualified as a pandemic. Yeah.
Yeah. Oh, that's interesting. Okay. So anyway, you were at home during the quarantine period. You took the test. You got into Peking University. You didn't have to take the test anyway. You studied biology, and then you decided to do your PhD. And how did you decide to do your PhD? Were you working in a lab, or were you just like, this is the natural next step for me?
In my grade, a lot of people are interested to study abroad. So I was one of them. In order to, it's also very competitive to go abroad, study abroad. I remember that year, Harvard only gave one position for the entire Peking University for our department for the biological science. So you can see how competitive, and consider how many, we all get a pretty good score. We come from all the states. So of course you need to have lab experience. So I joined a lab in my second year of college. And I just, I wouldn't say too much, but I just tell you, we work every day in the lab to 1 a.m.
Oh my God.
We don't leave the lab before 1 a.m. And then we come back next day, 9 a.m. to attend some class, and maybe around 3 our class finished, we come to the lab and work to the 1 a.m. And I did that for 3 years.
Wow. What was it like, productive work, or were you just like there? Like, was it like a FaceTime, like you had to be there, or were you actually working?
Actually working. It's like all biophysical. I was in a biochemical laboratory, so we keep expressing and purifying proteins at the time. So I remember I was talking to a friend, and he is right now assistant professor at the Peking University. He was my classmate. I was talking to him, and that was around 1 a.m. in the morning. I have the purified protein in my right hand. I have like a whisk on my left hand. That purified protein, I spent like 7 hours that day to purify. I was so tired, right, after class, and it's 1 a.m. So I was talking about, and I poured my purified protein down the drain. So I have to do it again.
Oh, so you just stayed overnight?
I don't think I did that, but I have to continue.
You had to do it later.
The next day, yeah.
Oh my God.
Some sleep. Yeah.
That's crazy.
Yeah.
And after all that, you still wanted to go do a PhD?
Yeah, you wonder. This guy must be crazy. Yeah.
But maybe it was like, usually, I mean, I think PhDs or labs aren't as intense in the States. Maybe not all labs, but I don't know, compared to that experience, it feels like US based research programs are like slightly better.
Yeah. So then I joined the Boyan lab in biomedical engineering department of Georgia Tech, right? The same lab. We spent time together. Yeah. And in Boyan's lab, our lab was one of the toughest lab in the department, because we have a lab meeting at 8:30 every day, right?
Yeah.
But that was not that bad.
I know. That's what I was like. I guess right? We're used to it.
Yeah, I was like, oh, I can handle this. Like, we get to the lab 8:30, and then we leave around 5-6, right? Usually, yeah. Sometimes later.
Yeah, usually you were in there like always after 5 though, but that's crazy. Okay, well, I didn't know your experience was like that, but that explains things. So then you did your PhD in biomedical engineering. Tell me what you studied in your PhD.
Yeah, I studied cell signaling in a type of cell that its function is to produce bone. It's called osteoblasts. I studied its response to vitamin D. I don't know. Maybe most of people are aware that vitamin D supplement is recommended by the doctors. and it's marketed online and it's help you to form bones to absorb calcium and give you a boost to your immunity. Even during COVID, some doctors say take some vitamin D supplement to help your immunity, because vitamin D actually has some regulatory role in the immune system as well. Yeah, I studied that signaling pathway, and I did also a little bit of, I think the majority of my work is focused on the cell signaling and biology, but I also did some tissue engineering, in the sense of we have this porous scaffold that we can grow a little bone graft in vitro. Yeah.
Cool. And you then decided to stay in academia. So at that point, you did you want to be a professor, or did you just want to do research? What was your goal after grad school?
Yeah, my goal is, my primary goal is always to try academic first, but with industry as my second choice. I actually went a couple of interviews with some company. I only did one interview for an academic postdoc position, which is turned out to be my lab for the next more than 10 years.
Okay, so from your PhD, so Georgia Tech, you went to Harvard for your postdoc, and you're still there after 10 years. So tell me about what you were doing at Harvard. What did you start doing, and what are the different, research topics you've worked on?
Yes, I joined the lab still sort of focused on cell signaling pathway, but the effector cell we study is macrophage. I don't know if have you watched the show in Netflix? It's about a macrophage. There is an animation about macrophage. Is the main character, and his girlfriend is red blood cell. It's a show about the immune system. It's really popular.
Is it for kids or what?
No, it's for everybody. Cool. But anyway, so macrophage is this guy that eat the dead cell and the pathogens, and it's our first line of defense against the infection. Also for homeostasis, tissue maintenance. We study glycoprotein on the macrophage surface. It's important in the inflammation resolution, which means how, we have a pimple, then we have redness, heat, pain, inflammation, and then eventually it goes away. That process of inflammation resolution is macrophage come to clean out all the wound and dead cell, and then they leave. That immigration of macrophage is what I was originally interested in. So I spent a couple of years study that. So my research project is a little spread, and then I got involved in another project. It's quite cool. It's a coating solid implant with a liquid surface. So then the idea is being, solid is always subject to biofouling, right? If you have implantation and your body gonna respond to it, trying to deposit things to it, trying to wrap it around, trying to use fibrosis to form a capsule, or even in the worst scenario, you got an infection, the bacteria start to interact with the implants, right? That's the major cause of implant failure. The idea is the liquid attached to nothing, right? Mm-hmm. The liquid, if you look at the surface of a lake, is already so pristine. So, and we didn't use just any liquid. We used a liquid that's called a perfluorocarbon. It's similar material of no sticky pan. Okay. So for cooking, and sometimes you saw the crazy commercials, a guy gonna pour a lot of a color to the coat of another guy and everything will gonna fall off. That's usually also perfluorocarbon coating. So we use a liquid format or perfluorocarbon to code a scaffold and to show how it can resist infection in vivo. So I studied that for probably a year, and then I got into another study. Is like it's a drug development project to focus on small molecules as immune modulators. The big idea being generally the anti-inflammatory strategy is pretty harsh, right? Either you give it steroid to suppress your immunity, or you have an immunosuppressant, which is even harsher, or you have a COX-2 inhibitor, which inhibits the generation of those inflammatory lipids. But it's usually, and the newer development is those cytokine antibodies, right? Anti-TNF-alpha antibody, anti-IL-1 beta antibody. But those works like on and off. It's trying to shut off pretty halves of the immune reaction. And that's usually associated to some level of side effect, including susceptibility to infections, right? You against inflammation, maybe shut your immunity down, and you are susceptible to infections. So those smaller molecules we are trying to develop it work as an immune modulator. It doesn't shut any major immune cascade down, but they tweak the kind of cytokine immune cell express. So the particular receptor I worked with is called the aryl hydrocarbon receptor, which is pretty cool. Is the host uses a sensor to interact with our environment. If you don't have that expression, the host lose a bunch of different type of immune cell. They wouldn't generally even develop to start with, and then the host is subject to a lot of infections. And the nice thing about that receptor is it's very druggable. It has a pretty versatile binding pocket. It can adopt a lot of small molecules. So if you design it well, you can avoid some of the side effects, and then you can train and tailor the immunity to perform in a way that you want it to perform, just similarly to how the environment interact with the host.
Okay.
And I'm also involved in another current project where we're trying to deliver genome editor in vivo. So genome editor is in general defined as an enzyme that can modify DNA, right? The really hot ones right now is called CRISPR, right? A lot of people heard about it. It has some really big buzz, and a lot of very successful biotech company is founded, and the Nobel Prize was awarded to this technology recently. So yeah, we're also trying to deliver the enzyme in the CRISPR technology is called Cas. So we're trying to deliver Cas protein with its guide RNA in vivo to go to the cell type that we are interested to edit the genome. So that is for the treatment of genetic disorders. I think I'm really interested in drug delivery or nanoparticle delivery space, where I'm currently developing for genome editor, but it can be used to just in general for intracellular protein and nucleic acid delivery, and then give you another level of controlling the cell signaling and the cell program and differentiation. And then you can apply that in vivo for treatment of immune-related disease, or you can apply it in ex vivo to modify cells and then you can use it as a cell therapy for transplantation. And then also continue to be interested in the small molecule immune modulator development.
Okay, and you're in Boston, so you're kind of at the epicenter of so many different great hospitals and great researchers and labs and stuff. Do you do a lot of collaborations with other people, or are you doing like clinical work at all?
Yeah, we do trying to collaborate as much as possible, and nowadays I think that's And maybe the only way to success is to bring a multidisciplinary team together, rely on other people's expertise, and also offer your own expertise.
Your favorite fast food restaurant?
McDonald's?
Yeah. Where if you had not got, if for some reason the PhD had not worked out, you could have worked there. And I think you wanted to work there.
Yeah, I like McDonald's. I like all the calories, after like a hard morning of experiment, right? You feel so tired, your whole body and mind is draining, and you think you have like another 6-7 hours to go in the afternoon. The only thing you think about is McDonald's with a big Coke.
Oh my God.
That is like supercharge, right? Supercharge me.
That is a great advertisement for them. But for like a very small.
All the calories. Sometimes I don't want the healthy food marketed as under 1,000 calories. How could that be enough? I want 2,000 for lunch alone.
Well, I don't know. I guess your body handles it, but not everybody's body can handle that many calories. So that's good to see you still like different. It.
I still do. I have a McDonald's very close to me, and there is this old gentleman work there. It's probably his 70s, and he had a tons. It's like it's McDonald's in the center of the hospital. You can imagine how many customers he knows.
Yeah.
And he remembers what I'm going to order.
Oh.
When I show up, he will just order.
What is your order? What's your go-to?
I have a seasonal shift. When the McRib is on the menu, I want the McRib.
I have never had the McRib. I should go try it. I don't
think it's on the menu right now.
Oh, okay. Well, I'll have to wait.
And McDonald's sometimes sell lobster roll in Boston. I don't know if they do it in other places.
That's cool. Is it actually good?
It's really good. It's really good.
Is it expensive? Lobster rolls are like $20.
I think in McDonald's it's 15, but it's not.
Okay, but that's
cheapest,
still
right? Yeah.
Okay, that's good. It's not like a $5 lobster roll where you're like, what is in this?
No. It's real lobster. You'll see the lobster meat. You don't see like identical red material. You see actually lobster meat.
That's so cool. I didn't know they did that. That's really neat. PhDizzle.
Having fun with everybody who do cool things.